Hello and welcome to a joint Innova Biosciences and Lumigen, a Beckman Coulter company webinar, Fast, Simple and Cost-Effective Immunoassay Development. Today's webinar will discuss immunoassay development, validation and sample analysis. This session will give a great overview of developing fast, simple and cost-effective immunoassays using the unique SPARCL proximity assay technology from Lumigen, alongside highly stable HRP conjugates developed with Lightning-Link labeling technology from Innova Biosciences. Topics covered will include an overview of SPARCL and Lightning-Link, fast and easy Acridan labeling, fast and easy antibody to HRP conjugation, and the benefits of the unique SPARCL proximity assay technology. Please note you can send in any questions you might have throughout this webinar session. Simply type your questions into the question box in your control panel and we will respond as soon as we can after today's session. So today we will have two speakers presenting. Our first speaker is Beth Wensley, Senior Scientist at Innova Biosciences. Beth works in our R&D team developing new antibody conjugation products, including our recently launched latex conjugation kits. Before coming to Innova, Beth was a postdoc at MedImmune, the large molecule division of AstraZeneca, working on antibody engineering. Prior to that, she did a PhD at the University of Cambridge, so has good experience of academia, large pharma and biotech. Our second speaker is Mark Cameron. Mark is a senior scientist at Beckman Coulter, Lumigen. Mark is responsible for supporting SPARCL customers and developing new applications for the SPARCL technology. Prior to joining Lumigen, Mark was a study director, associate director, and senior manager of the ligand binding assay lab at MPI Research, a contract research organisation specialising in drug safety and toxicology studies. Mark developed biomarker, pharmacokinetic, and anti-drug antibody assays in a GLP lab for pharmaceutical and biotechnology sponsors. Mark was a part of the Predictive Safety Testing Consortium, a collaborative effort that developed biomarker assays in support of drug safety. Prior to this, his experience at MPI Research, Mark commercialized antibodies, proteins, and research-use-only ELISA kits for assay designs and Cayman Chemical. Mark introduced to the commercial marketplace more than 30 ELISAs, including the first available ELISA to measure a phosphorylated protein, mitogen-activated protein kinase 3, also known as phospho-ERK. Mark has a wide range of biomarker and pharmacokinetic assay development experience, including colorimetric ELISA, Luminex, mesoscale discovery, Gyros, and SPARCL. Mark's latest publications provide recommendations for adaptation and validation of commercial immunoassay kits for biomarker quantification in drug development, makes recommendations for biomarker bioanalytical method development through the Global CRO Council, and details non-clinical biomarker assay development for vascular injury. It's time to move on to the presentation. Please feel free to send in questions through your control panel at any point. Thank you, and please welcome Beth.
Thank you for joining us for this webinar. We're excited to be talking in partnership with Lumigen. I'm going to start by giving you a quick overview of Innova Biosciences and our Lightning-Link antibody labeling kits. I'm then going to talk about traditional antibody conjugation and how our Lightning-Link kits can overcome many of the issues that can be associated with traditional conjugation. Finally, I'll talk about the various factors that need to be taken into consideration if you want to use our Lightning-Link kits, and finally look at some of the results and data that have been obtained using our Lightning-Link HRP specifically. Innova Biosciences is a biotechnology company that was founded in 2002 in Cambridge in the UK. Initially, we started making products for phosphate assay detection, although the company focus soon shifted into bioconjugation, and in 2006 we launched our Lightning-Link technology for antibody and protein labeling. I'm going to speak about this in more details later in the presentation. Following this, the company expanded into other protein and antibody labeling technologies to include gold nanoparticle labeling, our Innova Gold range of kits, which we launched in 2012, and then oligonucleotide conjugation kits in 2013. We're going to talk about Lightning-Link in this presentation, but if you have any further questions about our other labeling solutions, please do get in touch and we'll be happy to give you some more information about them. All of the technology that we have allows researchers to quickly and efficiently directly label our biomolecule without lengthy protocols, at the same time saving times and materials. If we take a bit of time to look at more detail at traditional antibody conjugations and hopefully explain why we have so many different antibody conjugation kits. Traditionally, being able to directly label your primary antibody with a label of your choice required experienced staff with immunochemistry knowledge and experience. Typically, there's a requirement for large amounts of your antibody, often 3 to 5 milligrams purified. So you need a lot of antibody to start, and this is to compensate for the large loss that often happens throughout the process. The separation steps and fraction collections that are needed after the conjugation reaction result in a large loss of the antibody, which really reduces the overall yield. The whole process takes quite a lot of time, and this can be very expensive. This is where Lightning-Link can really come in and make a big difference to your experiments. So Lightning-Link removes a lot of these problems associated with traditional conjugation. The kits are fast and the easiest to use method of directly labeling antibodies and proteins that are currently available. They only require 30 seconds hands-on time, and we have a really wide range of labels available, including enzymes, fluorescent proteins and dyes, tandem dyes, biotin, and streptavidin. So what is this technology that we have, and why do people that use it think it's so good? The beauty of it lies in its simplicity. If you look at this picture here, you can see you take your vial of Lightning-Link label, you simply add your antibody, you wait a specified amount of time, which depends on the kit used, then you add a quencher to stop the reaction, and then you have your labeled antibody. It's good to go, you can get on with your experiments. Because of this, the lack of separation steps needed, you retain all the antibody that you put into the kit. Unlike in a traditional conjugation, where the amount of antibody conjugated is quite small and you need to remove unlabeled dyes that haven't reacted, in Lightning-Link you don't have to do that, so you get 100% of your antibody back. Everything you put in the vial comes out of the vial and is good to use in your experiment. This is because we optimize the technology so that the protocol, when followed correctly, you get the correct ratio of label to antibody, and the majority, if not all of your antibody is labeled, so there's no need to wash away anything that's not been conjugated. We also have a quencher which stops the reaction running over completion, so you don't get any background from unconjugated label. Another major advantage of Lightning-Link versus traditional conjugation is the amount of label that can be conjugated. It's fully scalable from less than 10 microgram that you have of antibody that you can label yourself to the 1 gram scale for production manufacturer. And because it's a very specific process, there's very, very little batch-to-batch variability. So when you're scaling up, when you're repeating, doing the same thing over again, you get very similar results and it's easy to achieve. The technology is covalent, which ensures long-term stability of the conjugates. They can be stored for up to 18 months, so you can carry your experiments over time. Finally, as well as our standard traditional Lightning-Link kits, which have a 3-hour incubation kit period, we have a new range of Lightning-Link rapid kits, which only require 15 minutes. So you can get on with the rest of your experiments quickly, or even you can go home early, potentially. So this is why Lightning-Link is can be really good. So if you want to use it, what do you need with your antibody for it to work? The requirements are fairly simple. Your antibody, of course, needs to be purified, and it needs to be at a concentration of 1 mg per ml or higher, although this can depend on the label used. There are just a few buffer components that do need to be avoided due to the chemistry of the reaction. For example, azide, BSA, and tris inhibit the reaction. Although in very small quantities, they can be fine, and glycerol, when it's in your antibody, needs to be below 50%. These buffer requirements can be seen in this table on the slide, and any details and all details are shown within the protocol of each kit, which can be found on our website. So how does Lightning-Link compare? Lightning-Link's kits we use to directly label antibodies or proteins, and they can be used in a number of applications. However, a common misconception of direct labeling is that they can be less sensitive and produce weaker results than secondary detection. This isn't the case with our kits. We have lots of data to prove comparable, if not improved sensitivity using this technology. Here we've got some of the data showing Lightning-Link HRP across various applications to demonstrate what I'm telling you. This is a slide showing HRP Lightning-Link directly conjugated to an anti-human CD20 antibody versus a traditional secondary labeling technique. It's commonly thought, as I said, that direct staining is less sensitive than indirect staining. You can see here that using the directly conjugated antibodies, the results with Lightning-Link are very comparable to those that are produced with the indirect staining. This is another example slide. This is a Western blotting example. So this was using primary antibody, Lightning-Link labeled, or secondary detection. You can see from the amount of antibody that was used, the primary antibody working concentration, you actually needed quite a lot less of the Lightning labeled antibody than the unlabeled in order to detect a signal. And if you look at the band on the bottom on the Western blot analysis, you can see the bands are, it's a much clearer, much nicer band. You've got increased sensitivity and your picture's going to look a lot nicer, which can be especially useful if you want to publish it. This isn't always the case. We don't always see things that are better, but it does happen in quite a lot of times. Finally, this is a slide showing some ELISA data. This is a very sensitive CRP ELISA. You can see we're down in the picogram per mil CRP detection. This was using a calibration curve and spiking in some CRP into CRP depleted human serum to see what was happening if we got the same signal. And you can see that we've got a nice calibration curve, nice straight line, really clear results, small error bars, successful assay. We have over 60 references that support the use of direct labeling using our Lightning-Link kits. If you're interested, please take a look at our website, www.innovabiosciences.com/references.html, to see a list of these. While we're talking about HRP, I just wanted to quickly mention another HRP product we have, a HRP conjugate stabilizer. HRP activity can decrease for a number of reasons, and this can lead to all sorts of problems in assays and experiments. You can get inactivation of your HRP, you can get microbial attack, as can happen to all proteins. You can get your antibody that the HRP conjugate to denaturing, and you can get worse, these effects are worse at higher temperatures and at lower antibody concentrations. We sell a HRP conjugate stabilizer, which is called LifeXtend. This is a proprietary multi-component reagent system which protects HRP from these negative factors, including the ability to store the HRP at really, really dilute concentrations at the working concentrations you would use in assays, which can make it a lot easier to run and plan experiments. And now I want to hand over to Mark, who's going to be talking about the SPARCL platform.
Thank you, Beth. And hello, everyone. My name is Mark Cameron. I'm a senior application scientist at Lumigen, a Beckman Coulter company. Today, I will share information about SPARCL, a versatile and exciting technology from Beckman Coulter and Lumigen. SPARCL will change the way you develop and validate immunoassays and ligand binding assays. The use of Lightning-Link with SPARCL makes developing and validating immunoassays fast and easy. Today I'll give a technology overview about SPARCL, share how Lightning-Link and SPARCL work so well together. I'll highlight fast and easy immunoassay development. I'll give examples about immunoassay formats that can utilize SPARCL. I'll talk about SPARCL benefits and the use of SPARCL in a cost-effective strategy. A few words about Lumigen. Lumigen is a Beckman Coulter company located in southeast Michigan, near Detroit. We are a large facility established over 25 years ago. Lumigen makes substrates for Western blotting, ELISA, and clinical diagnostic instruments. Lumigen supports over 1 billion diagnostic tests each year by manufacturing luminescence substrates, typically used in clinical diagnostic instruments. If you've done Western blotting in your past and you've used ECL+, then you've used the Lumigen product. This ECL+ is referenced in thousands of publications. Lumigen also makes HyperBlue for direct hydrogen peroxide detection used in a single step. So let's move into SPARCL. SPARCL stands for Spatial Proximity Analyte Reagent Capture Luminescence. It's a novel homogeneous chemiluminescent technology. SPARCL is a proximity-based method. The core of the technology is Acridan. It's a small molecule that's triggered to release energy when associated with HRP and a hydrogen peroxide-based trigger solution. SPARCL is a reagent and a detection system that is plate-based. SPARCL is flash luminescence. This means you need a plate-based luminometer, and your luminometer must have injectors. And the BMG Omega series is a good example of luminometers that work well with SPARCL. So SPARCL is a tool for immunoassay development, validation, and sample analysis. The homogeneous part means there's no solid support involved, and by solid support we refer to a plate or a bead. There are no wash steps in a SPARCL assay because it's a proximity assay. It's convenient and reduces labor requirements for running assays because there are no coating of plates, no blocking of plates, no using of wash buffers, no using of proprietary discs, buffers, or plates. Assays run quite swiftly. Run times are 30 minutes or less in all cases. So let's talk about how SPARCL works. The key component is Acridan. It's a small molecule of about 700 Daltons that we use to tag key components of an immunoassay, in this case an antibody, by using simple NHS chemistry. So labeling with Acridan is fast and easy. Basically, we're going to dilute an antibody in a borate buffer, and we're going to reconstitute Acridan in an organic solvent and add the Acridan in solvent to our borate buffer diluted antibody. We're going to wait for 30 minutes, and at that point the antibody is ready to use. One of the nice features about SPARCL is there's no need for desalting or dialysis after labeling with Acridan. So in addition to Acridan as a key component for a SPARCL assay, HRP is also a key component. And we like to use Lightning-Link from Innova Biosciences for fast and easy HRP labeling. For HRP labeling, we're simply going to add a reagent to our antibody, transfer our antibody mixture to an HRP in a glass vial. We're going to follow the directions given by Innova and incubate that antibody with HRP at room temperature for 3 hours. At that point, the antibody is ready to use, and again, there's no need to desalt or dialyze after HRP labeling. So continuing on with our key components, we have an Acridan labeled antibody, an HRP labeled antibody, and our protein target. We're going to put those together in solution, and in this case, a single well of a 96 well plate is presented here. We're going to add our key conjugates and our standard or sample. We're going to incubate that for 30 minutes at room temperature. A note here about the volumes. These volumes are suggestions and a good starting point. SPARCL is a very flexible tool, so you can configure these assays to fit your particular needs. So after a short period of time, we have specific binding to our protein target with our conjugated antibodies, bringing them into close proximity. When we add our hydrogen peroxide-based trigger solution, we generate light from the association. In circumstances where there's unbound Acridan or HRP in the individual wells and the trigger solution is added, in this case we do not generate light because the key components are not in close proximity. So going back to our single well of a 96 well plate, the volume of reagents are in the bottom. After the short incubation period, 75 microliters of trigger is added by the luminometer and we generate light. That light is collected for 1 second per well. This is how the light is captured. It's flash luminescence, remember. SPARCL peaks at 0.15 seconds after injection of the trigger solution. We're going to collect this data in fast kinetic mode. We're going to integrate every 0.02 seconds for a total of 50 reads per second to generate this histogram looking graph. We're collecting area under the curve. This is relative light units versus time. And please note that time here is 0 to 1 second. Again, 1 second read per well, and collecting area under the curve. SPARCL requires a luminometer. The luminometer must have injectors. It's important that the luminometer is capable of reading the flash at the time of injection. Your luminometer may accommodate 96 or 384 well plates. Both are fine. We recommend white, solid bottom, low binding generic plates to run the assays in. BMG makes a series of luminometers called the Omega series. Please visit BMG LabTech for more information about their plate readers. You may also have instruments from molecular devices, the Spectrum XL, for example, or from Berthold, PerkinElmer, and other companies that work well for SPARCL. Please contact us to help you determine if your plate reader is configured to run SPARCL assays. A couple words about the injectors. These are really important. Injectors are pumps housed within the body of the plate reader. They can be syringe or other type of pump. These injectors are programmable to deliver precise volumes of trigger solution at a specific rate to each well of a 96 or 384 well plate. The use of injectors is essential. This is because of the reaction is so fast it cannot be done by hand. So let's move to the types of assays that you can use SPARCL with. And basically it's just about any type of assay that you can think of. I'll list a few here. We'll cover both sandwich and competitive immunoassays that are both quantitative and qualitative. A lot of high throughput agriculture folks use this for their testing, as do folks in the drug discovery, drug safety, and toxicology arenas for pharmacokinetic and anti-drug or immunogenicity assays. Biomarker assays of all types, monoclonal antibody development screening, host cell contamination testing, food contamination testing, protein-protein interactions, ligand binding assays of many types in many industries. SPARCL is a tool that can be widely applied. Please contact Lumigen if you have a question about assay design or application. So using a luminescent assay is much like using any other immunoassays. We can plot response, or in this case, relative light units versus concentration to generate a standard curve. This is a pretty typical standard curve for SPARCL. Please note the wide dynamic range and that we're using relative light units or area under the curve on the y-axis and concentration on the x. CVs are quite good with SPARCL. In this case, this is a pharmacokinetic assay, an assay with a wide dynamic range. This particular assay was run in triplicates. The raw values are plotted here in the center of the screen, and on the right-hand side in the red oval are the CVs. Non-zero CVs are all under 3%. The highest CV on this particular graph is 4.6. So you can expect excellent to exceptional CVs using SPARCL, and of course proper technique is essential when using a pipetter. If you're using automation, even better. You can get results even better than this work done by hand. Species and matrix. Basically any species and matrix is up for use with SPARCL. We have experience with human, rat, mouse, rabbit, dog, and non-human primate, both serum and plasma, as well as cell culture supernatants and buffers. Plant tissue homogenates. People grind up leaves, shoots, stems, and seeds, and use that as their matrix to evaluate for their particular protein in a SPARCL assay. So let's look at a couple of configurations, so how assays might look. This might be termed a sandwich assay, but of note, SPARCL does not use a solid support. On the left-hand side of this slide are key components: Acridan and HRP, conjugated antibodies, and our protein target. When we add these three reagents to a well and let them incubate, we have a specific immune complex formed, bringing our Acridan and HRP into close proximity. Competitive assays. SPARCL is well suited for designing and developing competitive assays. Again, it's configurable based on your need. In this particular example, we're looking at a cyclic AMP assay. The key components on the left: HRP, cyclic AMP, a small molecule, and an Acridan conjugated anti-cyclic AMP antibody. On the right is what this assay might look like if you wanted to develop it this way. Cyclic AMP is conjugated to HRP. Cyclic AMP in solution would bind to the specific anti-cyclic AMP antibody conjugated with Acridan and block its binding to the HRP conjugated molecule, thus preventing the proximity and limiting the light at the end. Pharmacokinetic or PK assays are very commonly used with SPARCL. In this assay format, we're measuring a drug, and in particular, we're using an antibody as a drug. And we're simply conjugating the anti-drug antibodies with Acridan and HRP and forming a sandwich assay with an antibody that is a drug. And again, when the specific immune complex is formed, Acridan and HRP are brought into close proximity. So we are releasing a new product in the next few weeks at Lumigen. This is a universal PK assay. This kit includes the SPARCL chemistry and the antibodies you need to develop PK assays for all human IgG subclass drugs. You have to supply the drug or the test article in this case. And this works well in species that include mouse, rat, dog, rabbit, and non-human primate. Using this technology and these antibodies, you can develop PK assays that are ready to validate in as little as 2 days. For more information, please visit Lumigen and fill out the Contact Us tab if you have more questions about that. Let's look at another format of anti-drug antibody format using SPARCL. In this case, we're trying to measure antibodies formed against drugs that have been dosed into animals or humans. We're going to label the drug in this case with Acridan and another fraction of the drug with HRP. We're going to mix those together with a sample. If the sample contains antibodies against the drug, a bridge will form. Now that's shown here on the right, where Acridan and HRP are brought into close proximity. Another example of where SPARCL is being applied is antibody development and screening. The process of monoclonal antibody development requires the testing of many hundreds of plates from tissue culture. And in this scenario, we're using an Acridan conjugated immunogen, an anti-species labeled HRP antibody, and our sample is in blue here on the left. On the right-hand side is what happens when you combine these key components into the well. We'll get a specific immune complex formed where the sample binds the immunogen and your anti-species antibody, HRP conjugated, will form the association of Acridan and HRP, allowing rapid and efficient testing of monoclonal antibody development, circumventing the traditional use of coated plates for ELISA screening. SPARCL assays can be miniaturized. Basically, any plate that goes in your luminometer or that you've been accustomed to using will work with SPARCL. It's a flexible format. Top left on the screen is a half area well, 96 well plate. Upper right are 384 well plates. These assays are great when you want to scale down and improve throughput and also conservation of rare samples. Cases where you simply don't have large volumes of samples. In this case, you can use a sample volume of 5 microliters. Also conserves on the expense around critical and valuable reagents. In high throughput screening, SPARCL offers a flexible format as far as timing. I mentioned earlier that we typically collect the SPARCL flash for 1 second. Here on the bottom is the typical flash and decay of the SPARCL chemistry from 0 to 1 second. In cases where you want to speed that up, you can collect your data from 0 to 0.7 or from 0.5 to 0.05 to 0.3 seconds, speeding up the process and getting many more samples run in a day. So speaking of throughput, Let's talk about automation for a few seconds. SPARCL is very automation friendly. This is because it runs in standard and generic microtiter plates of the 384 and 96 well plate formats. There are limited pipetting steps in a SPARCL assay. You can add your antibody mixture in one step and your samples and QCs in another. Let those incubate in generic plates with generic data acquisition and software analysis packages. It's high throughput. If your lab is Watson LIMS enabled, go ahead and use that technology for data acquisition and analysis. BMG makes luminometers with 50 plate stackers. This allows stacking plates on the luminometer and walking away. At 1 second read per well, you can read an entire plate in 2 minutes and certainly easily run 50 plates in a couple of hours. So immunoassay design and SPARCL. SPARCL is a highly flexible tool and easily configurable to suit your needs. The key things to remember are the Acridan and the HRP. They must be in your assay design somewhere. And the trigger solution is a hydrogen peroxide based. That has to be in your assay design. It's conveniently added by the luminometer. If you have favorite reagents you like to use, you can go ahead and use those. You can make your own conjugates if you need one. For example, if you're using biotinylated reagents, you can use a streptavidin Acridan conjugate. If you're using anti-tag antibodies, you certainly can go ahead and label those with Acridan, HRP, or biotin, depending on what your need is. If you like particular anti-species antibodies, go ahead and use those. You might need to label those with HRP or Acridan, but labeling is fast and easy. So an example of the flexible format with SPARCL, in the case where you like to use biotinylated antibodies, we're going to go ahead and do that. We're going to simply follow with a streptavidin HRP conjugate, allowing the immune complex to form specifically and bringing our Acridan into close proximity with HRP. When the trigger solution is added, this will generate light. SPARCL is great for those of us who are pressed for time. SPARCL greatly reduces and compresses your timelines. So as an example, antibody labeling and screening can be completed on day 1. Labeling in the morning using Lightning-Link HRP from Innova Biosciences and Acridan from Lumigen. You do best pair determination in the afternoon. Again, there's no blocking, coating, or washing of plates. On day 2, you can move to basic assay development tasks like titration, dynamic range determination, QC placements, and minimum required dilution determinations. On day 3, you can move to advanced assay development tasks, such as accuracy and precision, dilution, linearity, and parallelism. Core validation can be finished at the end of day 4. 6 accuracy and precision runs, including dilution, linearity, and parallelism, if that's your thing. Considering the short assay runtime, 30 minutes or less, you can certainly run 6 to 12 individual runs per day using the SPARCL format. So considering there's no coating or blocking of plates, SPARCL offers significant labor and time savings. Also offers a 30-minute or less assay runtime. There's no washing steps, no adding of substrate or read buffers, no waiting for color develop in the case of colorimetric assays, no use of acids or stop solutions in the case of colorimetric assays. There's an immediate generation of signal when the SPARCL trigger is added. This allows you to read an entire plate in 2 minutes with walkaway automation possible when stackers are used with the luminometers. The only special requirement needed for a SPARCL assay is the luminometer with injectors. Many of you already have luminometers. Some of you have the injectors on those luminometers. The upgrade of your current luminometer with injectors is certainly possible. It's interesting that the cost of a luminometer is often less than the service contract for some of the bigger box platforms. So if you're considering one of those big platforms, the service contract is often more than what a luminometer actually costs. Disposables. So it's a simple and easy format. So the assay buffers are also simple. You're going to make your own or purchase your favorite. We recommend starting with a PBS tween or PBS BSA, for example. Running generic white 96 or 384 well plates. No proprietary buffers, plates, or spinning discs. No wash buffers at all. There's a lot of labor saved in not having to stop and wash plates and add reagents every hour or two. So SPARCL is simple, easy to use, and cost effective. Please see the Lumigen catalog for the current offerings we have for SPARCL configuration at lumigen.com. There is a lot of technical support available to answer your questions. If you'd like to discuss a potential project with Lumigen scientists, please go to lumigen.com and use the Contact Us tab and fill out the form that's there. We can help you with your specific application. Will SPARCL work for you? We can design or review your assay design. We can make sure your luminometer is technically capable of running SPARCL assays and help you with the settings. If you don't have a luminometer, we can help you find a demo unit to test. We also can help with reviewing your data and your assay optimization during your development time. So as we start to wrap up, SPARCL is a flexible assay development tool that can be widely applied to numerous immunoassay formats. And these formats include sandwich, competitive, quantitative, qualitative, and high throughput applications, specific applications like biomarker assays, pharmacokinetic assays, anti-drug antibody assays. And a lot of folks are converting ELISA, MSD, and Gyros to SPARCL formats to save on cost of reagents, disposables, and labor. SPARCL assays provide performance, a wide dynamic range, great sensitivity, low minimum required dilution, short assay runtimes, conservations of reagents and samples. SPARCL is simple, fast, and easy. SPARCL enables a cost-effective approach. There's cost savings realized in virtually every aspect of assay development, validation, and production, including labor, capital equipment, disposables, service contracts, and buffers. SPARCL offers a simplification to a process that in some cases become very cumbersome and expensive. Using SPARCL, you have control of your assay. Again, if you are in pharmacokinetics, drug safety, we are about to release a universal PK assay that works well for any human IgG drug and any preclinical species. Please use lumigen.com and the Contact Us tab and fill out the form with any questions that you have. For our colleagues in Europe, we'd like to suggest that you use Direct, www.direct.com, or info@direct.com for email inquiries. You may also contact us at SPARCL@Beckman.com or at info@Innova Biosciences.com. This concludes my presentation about SPARCL. Thank you for attending this webinar. I hope you found the information useful. Again, please feel free to contact us with any questions or concern. And now let's go back to Clau for her final and closing remarks. Thank you.
Well, thank you, Mark. Thank you, Beth. We hope you enjoyed today's webinar. If you didn't get a chance to ask us any questions, then please note you can reach us at info@innovabiosciences.com or sparkle@beckman.com, and we will get back to you as soon as possible. Additionally, if you would like to receive a PDF copy of the presentation or a link to the video recording of the webinar, please contact us at those same email addresses. Please bear in mind that this webinar is recorded and we will upload it to our YouTube channel in the next few days so that you can always come back to it. You can find our channel at youtube.com/Innova Biosciences. Thank you for joining us for this webinar today, and we hope that you enjoyed the webinar.